Good morning, one and all. Today's topic is the antimicrobial agent. I am going to brief about antimicrobial agent. What is antimicrobial agent? Any substances that can kill or stop multiplication of microorganisms. Antibiotic. Then what is the difference between antimicrobial agent and antibiotic when we call it antibiotics? Antibiotics is substances produced by microorganism. That substances can kill or inhibit the other microbial growth. Antibiotic are naturally occurring microbial product, including some modification, main chemical modification can be done. Some compound derived from synthetically by using synthesis molecule. So that is called chemotherapeutic agent. Some of the antibiotic is a slight modification. We isolated from natural source, example for plant. So that compound can be modified, slight modification, that is called semi-synthetic compound. So here, how you can see the antibiotic activity. So here this slide shows very clear picture about how this antibiotic can inhibit the microorganism. Here you can see, first we need to inoculate the bacteria on the surface of the plate. Then we have to place the antibiotic disc, that paper disc incorporated with the respective antibiotic. So we have to place on the plate, then incubate for 18 to 24 hours. Then after 24 hours, we have to measure the zone of inhibition. The zone of inhibition indicate the activity of antibiotic effectiveness. So then antibiotic are selective toxicity. That means the drug should not affect the host, always toxic to the microorganisms. Then antibiotic can be characterized by their theoretical ratio. The highest dose of drug a patient can tolerate without having any side effect. Okay, so the minimal dose required for control of microorganisms. Then antibiotic can be classified based on the spectrum of action. The antibiotic may be bactericidal or bacterial-static. What is a bactericidal? Bactericidal, the antibiotic should kill the microbes, but bacterial-static may or may not kill, mean it should inhibit the growth of or inhibit the multiplication of the bacteria. So then next classification, the antibiotic is a spectrum of action. Some of the antibiotic we can call it as a narrow spectrum of antibiotic. This group of antibiotic, which is is active against one particular group of bacteria. Example for it act on only gram positive. It doesn't act on as a gram negative. Okay, some of the antibiotic is only action on only the gram negative bacteria. Example polymyxin. Only the gram positive bacteria is a bacitracin and vancomycin. Then broad spectrum. Other category of antibiotic called broad spectrum of antibiotic. This type of antibiotic, example for tetracycline, it can act active against gram positive as well as gram negative bacteria. Then mode of action, how this antibiotic is act on the bacteria. So antibiotic after entering the microbial cells attack one of the following 4 targets in the microorganisms. One is cell wall synthesis inhibitory antibiotic, another one is a cell membrane inhibitory antibiotic, and protein inhibitory antibiotic, and nucleic acid inhibitory antibiotic. Commonly we can see, so this picture shows very clear. So you can see cell wall inhibitory antibiotic, example for penicillin, cephalosporin, monobactam, and bacitracin, and the cell membrane antibiotic is a polymyxin. Then protein inhibitory antibiotic erythromycin and tetracycline, and nucleic acid synthesis antibiotic also. So now this cell wall inhibitory antibiotic group of antibiotic that is belonging to beta lactam agent. What is a beta lactam? This group of antibiotic, it has the molecule beta lactam ring in their structure. So the group of antibiotic beta lactam agent, penicillin, cephalosporin, monobactam, and carbapenem group of antibiotic. Those antibiotic always act on the cell wall. Okay, this is called penicillin, cephalosporin, carbapenem, and monobactam. Okay, so mechanism of action of beta lactam ring of antibiotic. This once is apply the antibiotic on the bacteria. So what happened? This molecule can bind with the penicillin binding protein. This protein located or situated between cell wall and cell membrane. So where this molecule can bind and act on the cell wall. So what happened once this molecule bind? So what happened is enhance the lytic enzyme action. So what happened? This lytic enzyme is this enzyme, the presence of penicillin, the autolytic enzyme function more action, extensively causing the cell lysis. So bacteria obviously will die quickly. So here, this is a penicillin binding protein. So this molecule can bind the penicillin binding protein. So what happens is bacterial cell wall get destructed like this. Okay. So then next group of antibiotic, the action mechanism of resistance. So okay, the penicillin, cephalosporin, monobactam and all, this is called cell wall inhibitory antibiotic. However, this antibiotic, it shows resistant, beta lactamase resistant. So synthesis of beta lactamase the bacteria can produce some of the enzyme called beta lactamase. This enzyme can cleave or destroy the beta lactam ring. So what happened? Obviously, this bacteria become resistant against the beta lactam ring group of antibiotic. Okay. So how this resistant happens? So the bacteria, gram-negative bacteria, always they produce beta lactamase enzyme. So upon the apply the antibiotic on the bacteria, what happens? This enzyme can cleave or destroy the beta lactam ring. So the bacteria get resistant. Other one is called the other mechanism of resistance. We can call it as mutations. Mutation happen in the penicillin binding protein itself. So what happened? This molecule may not bind with penicillin binding proteins. Obviously, bacteria shows resistant. Okay. So now next one is next category. We can say the protein inhibitory antibiotic. So this group of antibiotic is always they suppress the protein synthesis. The bacterial ribosomes is a 70S ribosome or sufficient differentiation from the mammalian 80S ribosomes. The antibiotic affecting the bacterial protein synthesis. So what happens is because of this antibiotic action, this bacteria may not synthesize necessary proteins. Essential proteins. So bacteria obviously they suppress. Antibiotic affecting the protein synthesis can be classified into two groups. One group is always suppress the 30S ribosome subunit. Another one is a 50S ribosome group of antibiotic. Okay. So first we see the 30S ribosome inhibition antibiotic aminoglycosides. So this one is they're causing misleading of the mRNA, resulting in wrong amino acid coding. So incorporation during the protein synthesis. That's why the bacteria may not synthesize the complete protein. So bacteria may not survive. So another one is called another group of antibiotic we can call antibiotic inhibit nucleic acid synthesis. So this role the following antibiotic we can call it nucleic acid synthesis inhibitory antibiotic. So what happened, this type of antibiotic, they affect the nucleic acid synthesis, they suppress the nucleic acid synthesis. So bacteria may not survive in presence of the particular antibiotic. Okay, thank you so much.